Good evening, viewers. This is welcome to MouShauk, and today we will start with the question answer of digestive system part 4. This will be ending the chapter. Okay, and before I start, I will request you to please do subscribe my channel if you have not subscribed yet, and if you are a new viewer to my channel. So please stay tuned, and if you have already been subscribed, then please stay tuned and watch my videos that will help you in your studies, okay, preparing for your exams, okay. And if you are liking the video, press the like button, share with your friends so that they can also be beneficial from these videos, and do meditate before you start your day. So we will also start with a short meditation, and then we will start with the question answers, okay. So for this, let us concentrate at the point of light. Think that light is falling through the source of energy that is falling through, falling between your eyebrows. Your soul is there. Your soul is being energized with that light. You're getting power. You're getting interest. You're becoming very calm, quiet. You're feeling this. You're feeling a lot of things. You're relaxing. You're becoming happy. Feeling that you're being held with everything that you're doing. Okay, now, whatever that God you want to do, please let it be successful. To the end of the day, what you have done, you have to use so much. Okay? So let us end here and start with a question answer series. So for that, first question for today is, what is the composition of feces? So our feces contains 75% of water and 25% of solid matter. Okay, it consists of the solid matter itself, this 25% solid matter that is also divided into four parts. That is 30% of dead bacteria, 10 to 20% of fat, 2 to 3% of proteins, and 30% of roughage. Now, what is the reason of the bad odor of the feces? We find a very bad odor of our feces. Why it is so? It is due to the bacterial action. Now, what is done by the assimilation of food? Okay, this assimilation of food is conversion of the dissolved digestive food, sorry, absorbed digestive food into the body material. So our food which we eat has to be converted into body material so that the body can use it for getting energy and for doing different functions of our body, bodily functions, that is. Okay. So for that reason, the food which we take in, that has to be converted into the body material, and that is called assimilation of food. Okay. Now, how the foods are transported, okay, which are digested and absorbed by the gut. We know that the food is being digested by our alimentary canal or the gut, and then it is absorbed by it. Okay. So in two ways, these food is transported to our cells. Okay. So what is that? First is through hepatic portal system and through lymphatic system. So two systems are there which transports our food. That is hepatic portal system and lymphatic system. Okay. Now, name the nutrients which are transported to the liver by the hepatic portal vein. Okay, so hepatic portal vein transports simple sugars, amino acids, vitamins, and minerals. Okay, from the intestine to the liver. It is converted. It is transported by the hepatic portal vein. Okay. Now, how are the glucose or simple sugars assimilated? First, what happens? Excess of glucose. We take carbohydrates, so that is converted into glucose. Simplest form of carbohydrate is glucose. So for that reason, it is converted to glucose so that the cells can absorb it. So excess of the glucose or other simple sugars are converted into insoluble glycogen by the liver from the blood. It's taken away from the blood. So what happens? The intestine, after being absorbed by the intestine, it gives to the blood. Okay, so this blood from if the blood has more, excess of glucose or other simple sugars, that excess of glucose and simple sugars are taken by the liver, are converted by the liver into glycogen, and they are temporarily stored by the process of glycogenesis. So by the process of glycogenesis, this liver functions in this way. That is, it converts excess glucose or simple sugars, other simple sugars into glycogen and temporarily stores it. Okay. Then when it is required, when the body requires glucose, when the body requires energy, then what happens? This glycogen is reconverted into glucose. Okay, by the liver again, and by the process called glycogenolysis. Okay. Glycogenolysis. Glycogen is broken down. Lysis means broken down. So glycogenolysis. Okay. And the first one is glycogenesis. It is glycogen is formed. Okay. And here glycogen genolysis. Here it is broken down and return to the blood circulation. Now then, glucose is used by the body tissues. When the glucose is used by the body tissues for getting energy. That is during cellular respiration and during the synthesis of certain compounds. When certain compounds are synthesized or cellular respiration occurs, then what happens? The body tissues requires energy, which is given by the glucose. Okay. Now, how are amino acids assimilated? The end product of protein, the simplest form of protein is amino acids. Okay, so the proteins are broken down into amino acids so that the body can absorb. Okay, now the amino acids, they are the building blocks of protein, or the simplest form of protein is amino acids. Okay, they cannot be stored, so they circulate in the body. In the blood circulation, they remain. Excess of them is broken down by the liver by the process of deamination. Okay, excess of protein, if the blood has more of protein, then that excess is broken down by the liver by the process of deamination, where the amino group or the nitrogen group, nitrogen containing amino group, that is removed from the protein part and converted to urea. Okay, and that urea is removed from the body by excretion. Okay, when we excrete, that urea part is removed from the body. And the remaining part of the protein, that is amino group, is removed first. That the remaining part of the protein is converted to glucose, which is then utilized to form the energy. Okay, this is how the amino acids are assimilated. Now, how are fatty acids and glycerol assimilated? Now, the fatty acids and the glycerol are the simplest form of fats. Okay? That is, when the fats are broken down, the end product is fatty acids and glycerol. Or when we, they are the building blocks of fat, you can say. Okay, the fatty acids and glycerol, which are absorbed by the alimentary canal, are transported through the intestinal villi and the lymphatic system. They are not converted. They are not transported by the blood circulation directly. They are transported by the lymphatic system. In the villi, we have the lacteals, which is the lymphatic vessels. Okay, so through that lymph vessel, through that they enter the lymphatic system, and then they move into the large veins, which carries them to the heart. Okay? So the thoracic duct of the lymphatic system empties into the large veins, okay, which carries the blood to the heart. Now, some of the fats are used by the synthesizing for synthesizing certain compounds in the body cell. Now, these fats, some of the fats are used for synthesizing certain compounds in the cell, body cells. Okay, the excess quantity of the fat. If the blood circulation has more of fat, then that is deposited in certain under the skin, below the skin as subcutaneous tissue. Okay, subcutaneous fat it is, and also they are deposited around certain visceral organs in small quantities. Then, which is the largest gland of our body, and what is its average weight at its location? The largest gland is the liver, which is reddish brown in color, and its average weight is 1.5 kgs. It is located in the upper right side of the abdomen, just below the diaphragm. The thoracic and the abdomen is divided by the diaphragm. Okay, that will just below the diaphragm on the upper right hand side, we have the liver. Now, what are the functions of the liver? Main function is production of bile. Okay. Now, other functions are also there. That is regulation of the blood sugar, deamination. We have studied just now only that two functions. That is blood controlling of the sugar, glucose level inside the blood, and another is amination, deamination process, which is done to remove the excess protein from the blood. That is deamination producing urea and sugar. Synthesizing fetal RBC when fetus is in the womb of the mother, then that time the fetal RBC is synthesized by the liver. Then storage of iron, copper, and several vitamins, etc. is stored. Liver stores iron, copper, and several vitamins. Then detoxifies the drugs and alcohol. Okay. Then production, it produces fibrinogen and prothrombin, which are utilized for blood clotting. Okay. Then it produces heparin, allergic reactions. No? So it produces heparin. Then production of heat by cellular metabolism. When cell metabolization takes place, then it produces heat. The liver produces heat. Regulation of blood volume by acting as a temporary store of excess water. Excess water is stored in the liver, and it regulates the blood volume. When the blood becomes thick, it releases the water so that the blood becomes fluidic. Okay, and when it is more of fluid, the blood is more of fluid, more liquidy, then it takes away the water from the blood and stores it. Then excretion of toxic and metallic poisons. Okay, some toxic or metallic poisons, if they are present, it excretes, it has an excretion. Then it destructs the dead RBC. Okay, producing bile. Okay, that bilirubin color. Okay, the color is comes from this dead RBC by breaking down of the dead RBC. Now, how is blood sugar level regulated by the liver? Now, liver, we have studied earlier only that liver returns the excess glucose, which retains the excess glucose, which is received as a product of carbohydrate digestion. When carbohydrate digestion takes place, it receives excess glucose. The blood receives excess glucose. Okay, that excess glucose is converted by the liver into glycogen and releases the glucose. Releases and it reconverts the glycogen into glucose when needed into the blood circulation. One is formation of glycogen by glycogenesis and releasing the glucose by reconverting the glycogen into glucose, that is glycogenolysis. So these two processes help performed by the liver to control the blood sugar level. Okay. Then why liver is an important organ in the post-mortem examination? Because it provides a dependable clue to suspect cases of poisoning through food. If one gets poisoning through food, that poison gets stored in the liver. So from if we examine the liver, we can get the, I mean, if it is positive, then if the person is poisoned by the from the food, then we can get the result positive. Okay? Then how can we prepare a suspension of starch? If 1 gram of starch is added to 100 ml of water in a beaker and heated, then we can prepare a suspension of starch. So 1 gram of starch is 100 ml of water. Okay, then heated it. Then it produces suspension of starch. Then how can we show that the action of saliva on starch does not give a positive starch test? Okay. We know that positive starch test means if we add iodine to starch solution, it will turn blue black. But if we add saliva in it, okay, after some time we will see that the starch solution, which is a turbid type solution, that will change into clear solution and it will not uh, show this, it will not turn blue black after addition of iodine. Okay, so for this, the test is like this. The first test tube, we take a starch suspension and divide it into 3 test tubes. In the first test tube, we add iodine solution. Second test tube, we test it for sugar. Okay, and in the third test tube, we add same quantity of saliva in it. Then we keep it for 15 minutes in our hand so that we keep it in warm condition, or we can also put it in a beaker, which is around 38 degrees centigrade. In the beaker contains water, which is about 38 degrees centigrade, and in that we put the test tube so that it remains warm. After 15 minutes, we will divide this third test tube content into three parts. I mean, 2 parts. First, we will test it for sugar. Another, we will test it for starch. So when we are testing for sugar, we will see that it turns positive. That means after addition of saliva, it has changed into sugar. The starch has changed into sugar. Okay. And if we, in the second part, if we test it for starch, that is by adding iodine solution, it will turn negative. That means the starch is not there now. Starch has been digested by the saliva. Saliva contains what? Saliva contains enzyme, starch digestive enzyme, that is amylase or thymine. Okay. Now, what happens when pepsin acts on egg white? So, egg white is a protein. Now, on which pepsin acts, but one thing is there that peptin cannot act directly on protein. Protein has to be in acidic medium before pepsin acts on it. Okay, it will not be in alkaline medium. It has to be some acidic medium. So pepsin, first we take the protein, that is egg white, and pepsin will digest it. Okay, so when pepsin acts on egg white, it digests it, but the egg white has to be mixed with hydrochloric acid to make it acidic. The medium should be acidic. Okay, so we add some acid in the egg white so that it becomes acidic. And now we add some pepsin in it. We will see that the protein will digest the pepsin will digest the protein egg white. It will become a clear solution. And also it requires a 35 degrees temperature for the digestion of the protein. Okay. Now what happens if the egg white suspension is made alkaline with a mild sodium bicarbonate solution and kept at 35 degrees centigrade? Will pepsin digest this egg white? No. First it is no. Then this egg white solution, this suspension, because this egg white suspension is made alkaline by mild sodium bicarbonate solution, and pepsin cannot act in an alkaline medium. Okay, it has to be an acidic medium where the pepsin can act on the protein. Okay. Now mention a test for glucose. A little glucose is added to a blue-green solution, which is known as spelling solution, in a test tube. Okay. with the test tube where the solution is tilted and then it's heated. Okay, till the solution starts bubbling. We can see the bubblings in that test tube. Till that, we will heat it. We will not overboil it. Okay. Now the color of this spelling solution, that is blue-green solution, will turn to deep brick red precipitate. Solution will turn precipitate and the color will change from blue-green to deep brick red color, deep brick red color, indicating that the presence of glucose, which indicates that there is glucose. Okay. Now, what is the test for sucrose? Here, sucrose is also a sugar, but it's a disaccharide. Okay? Now, glucose is a simple sugar monosaccharide, but in this sucrose, we will first add dilute hydrochloric acid in it, and then we will neutralize with sodium bicarbonate solution. Okay. Then we will add this, in this solution, we will add some pairing solution. Okay. Now, we will see that this will test now positive for sucrose. Okay. Now this sucrose will test positive. I mean that this sucrose we will be we can confirm that the solution is a sucrose solution. Okay. Next, what is the positive test for starch? Another thing is that in the previous question, if we do not add HCl acid or neutralize with sodium bicarbonate solution, then this test will not come to positive. It will not show sucrose. Okay. What is the positive test for starch? First, a starch solution is made with a little starch powder in a test tube with some water and then boiled. Then when cold, a few drops of iodine solution is added. The starch solution will turn blue-black, which indicates the presence of starch. Okay, positive test for starch positive test for protein. When piece of hard-boiled egg is taken in a test tube and few drops of concentrated nitric acid is is added enough to immerse the sample of food. There's such amount of nitric acid is given so that egg white is completely immersed in it. Okay. Then the test tube is gently heated. Now, after cooling, we add some ammonium hydroxide. Now, it is observed when we add nitric acid, a white precipitate is formed. Then when we heat the test tube, we will see that white precipitate will turn to yellow. Now, when we cool it and add ammonium hydroxide to it, then that yellow precipitate will turn to orange precipitate, which shows that it's a protein. It's egg white, but the food sample is protein. Okay. Now, what is the test for fats and oil? A piece of groundnut or walnut or a piece of butter is rubbed on a plain paper. The spot rubbed, whether the butter or the walnut or groundnut is rubbed, that place becomes translucent. Okay, when viewed under light, you can see that it is become translucent. So this indicates that the product which we are using, that sample which we are using, that contains fat or oil. Okay. So this much is for today, and this is the end of the chapter Digestive System, Transparent Syllabus. We will start with the next chapter, that is locomotion and movement of this chapter. Okay. So for that, stay tuned and do observe my test videos as well as question answer videos that will prepare you in all respects the chapter. Okay, and you can easily solve all of your questions. Okay. So please do concentrate, and you can get all the links of this chapter in the description box. Okay. So thank you for joining, and do meditate before you start your day. Thank you.